Hi class, this is your instructor, Skylar Huff. We're now in Chapter 21, The Evidence for Evolution. As briefly discussed before, when Darwin proposed his revolutionary theory of evolution by natural selection, little actual evidence existed to bolster his case. Instead, Darwin relied on observations of the natural world, logic, and results obtained by breeders working with domestic animals. Since his day, however, the evidence for Darwin's theory has become overwhelming. The case is built upon two pillars. First, evidence that natural selection can produce evolutionary change, and second, evidence from the fossil record that evolution has occurred. The whale skeleton you see here is the Vogel whale, which is the oldest whale fossil found in North America. It's 40 million years old. The pelvic bones and hind limbs reveal a link between land animals and whales. In addition to the evidence from studies of natural selection and fossils, information from many different areas of biology, fields as different as anatomy, molecular biology, and biogeography is only interpretable scientifically as being the outcome of evolution. So let's get to our first example with the beaks of Darwin's finches. As you learned in Chapter 20, a variety of processes can produce evolutionary change. Most evolutionary biologists, however, agree that Darwin's thinking that natural selection is a primary process responsible for evolution. Although we cannot travel back through time, modern-day evidence allows us to test hypotheses about how evolution proceeds and confirms the power of natural selection as an agent of evolutionary change. This evidence comes from both the field and the laboratory, and from both natural and human-altered situations. Darwin's finches are a classic example of evolution by natural selection. When he visited the Galapagos Islands off the coast of Ecuador in 1835, Darwin collected 31 specimens of finches from 3 islands. Not being an expert on birds, Darwin had trouble identifying the species, believing by examining their beaks that his collection contained wrens, grosbeaks, and blackbirds, all birds with which he was familiar from his native England. Upon Darwin's return home, ornithologist John Gould informed Darwin that the species he had collected were not closely related to different English birds, but rather they were closely related to a group of distinct species, all similar to one another except for their beaks. In all, 14 species are now recognized. The diversity of Darwin's finches, as illustrated here in the figure you see, figure 21.1. The finches feed on seeds that they crush in their powerful beaks. So again, I say the ground finches, they are the ones who feed on seeds that they crush with their powerful beaks. Species with smaller, narrower beaks, such as the warbler finch, eat insects. Other species include fruit and bud eaters, and species that feed on cactus fruits and the insects they attract. Some populations of the sharp-beaked ground finch even include vampires that sometimes creep up on seabirds and use their sharp beaks to pierce the seabird's skin and drink their blood. Perhaps the most remarkable are the tool users, woodpecker finches that pick up a twig, cactus spine, or leaf stalk, trim it into a shape with their beaks, and then poke it into dead branches to pry out grubs. That correspondence between the beaks of the finches, or the finch species, and their food sources suggested to Darwin that natural selection had shaped them. In The Voyage of the Beagle, written several years after his return to England, Darwin wrote, "Seeing this gradation and diversity of structure in one small, intimately related group of birds, one might really fancy that from an original paucity of birds in this archipelago, one species has been taken and modified for different ends." Darwin's observations suggest that differences among species in beak size and shape have evolved as species adapted to different food sources. But can this hypothesis be tested? In Chapter 20, you read about the theory of evolution by natural selection, requires three conditions be met. Phenotypic variation must exist in the population. This variation must lead to differences among individuals in lifetime reproductive success. And then lastly, phenotypic variation among individuals must be genetically transmissible to the next generation. The key to successful testing of Darwin's proposal proved to be patience. For more than 40 years, starting in 1973, Peter and Rosemary Grant of Princeton University and their students tested the medium ground finch on a tiny island in the center of the Galapagos called Daphne Major. These finches feed preferentially on small tender seeds produced in abundance by plants in wet years. During long periods of drought, the birds resort to large, drier seeds, which are harder to crush. The Grants quantified beak shape among the medium ground finches of Daphne Major by carefully measuring beak depth, and that's the height of the beak from top to bottom at its base, on individual birds. Measuring many birds every year, they were able to assemble, for the first time, a detailed portrait of evolution in action. The Grants found that not only did a great deal of variation in beak depth exist among members of the population, but the average beak depth changed from one year to the next in a predictable fashion. During droughts, plants produced few seeds, and all available small seeds were quickly eaten, leaving large seeds as the remaining food source. As a result, birds with shorter, deeper, more powerful beaks survived better because they were better able to break open these large seeds. Conversely, in particularly wet years, plants flourished, producing an abundance of small seeds. As a result, birds with long and shallow beaks were favored, and beaks became pointier. Could these changes in beak dimension be evidence of evolution by natural selection? If so, the variation among individuals in beak size must be genetically based. An alternative possibility might be that the changes in beak depth do not reflect changes in gene frequency, but rather are a simple response to diet. For example, perhaps crushing large seeds causes a growing bird to develop a larger beak. To rule out this possibility, the Grants tested for genetic basis to beak size variation by measuring the relationship of parent beak size to offspring beak size, examining many broods over several years. The depth of the beak was very similar between parents and offspring, regardless of environmental conditions. And you can see this shown in figure 21.2b, shown here on the right. So suggesting that the differences among individuals in beak size reflect genetic differences. This result satisfies the condition number 3 that mentioned above, and therefore it indicates that year-to-year changes in average beak depth represent evolutionary change resulting from natural selection. When the environment changes, natural selection often favors a trait that previously wasn't favored. One classic example concerns the peppered moth, Biston bitulera. Adults come in a range of shades from light gray with black speckling, hence the name peppered moth, to jet black, those that are melanic. Extensive genetic analysis has shown that a moth's body color is a genetic trait that reflects different alleles of a single gene. Recent molecular genetic studies have demonstrated that all black individuals are the descendant of a single mutation. This dominant allele was present but very rare in populations before 1850. From that time on, dark individuals increased in frequency in moth populations near industrialized centers until they made up almost 100% of these populations. Biologists also noticed that in industrialized regions where the dark moths were common, the tree trunks were darkened almost black by the soot of pollution, which killed many of the light-colored lichens on tree trunks. Why did dark moths gain a survival advantage around 1850? In 1896, a British amateur moth collector named J.W. Tutt proposed that what became the most commonly accepted hypothesis explaining the decline in light-colored moths. He suggested that peppered forms were more visible to predators on sooty trees that have lost their lichens. Consequently, birds ate the peppered moths resting on the trunks of trees during the day. The black forms, in contrast, had an advantage because they were camouflaged. And please see figure 21 point. Although Tutt initially had no evidence, British ecology Bernard Kettlewell tested the hypothesis in the 1950s by releasing equal numbers of dark and light individuals into two sets of woods, one near heavily polluted Birmingham and the other in unpolluted Dorset. If Tutt was correct, then the dark moths should have survived better in the Birmingham woods and the light moths in the Dorset woods. Kettlewell then set up lights in the woods to attract moths to traps to see how many of both kinds of moths survived. To identify the moths he released, he had marked the release moths with a dot of paint on the underside of their wings, where birds could not see it. In the polluted area near Birmingham, Kettlewell recaptured only 19% of the light moths, but 40% of the dark ones. This indicated that the dark moths survived better in these polluted woods, where the tree trunks were dark. In the relatively unpolluted Dorset woods, Kettlewell recovered 12.5% of the light moths, but only 6% of the dark ones. This result indicated that where the tree trunks were still light-colored, light moths' survival was twice as great as that of dark moths. Kettlewell later solidified his argument by placing moths on trees and filming birds looking for food. Sometimes bird actually passed right over a moth that was the same color as its background. Recently, an enormous 6-year study involving the release of nearly 5,000 moths confirmed Kettlewell's findings. Conducted in an unpolluted forest, the study found that dark-colored moths disappeared at a rate 10% higher than that of light-colored moths. In addition, direct observations of 250 feeding events revealed that dark moths were captured by birds substantially more often than light-colored moths. And this shows you what those moths look like. On the left, you can see the dark or melanic variant of the moth is more visible to predators on unpolluted trees, whereas the light-peppered moth is more visible to predators on bark blackened by industrial pollution. In industrialized areas throughout Eurasia and North America, dozens of other species of moths have evolved in the same way as a peppered moth. The term industrial melanism refers to the phenomenon in which dark individuals come to predominate over lighter ones. In Great Britain, the air pollution that promoted the industrial melanism began to reverse following enactment of the Clean Air Act in 1956. Beginning in 1959, the Biston population at Caldy Common outside Liverpool has been sampled each year. The frequency of melanic dark form has dropped from a high of 93% in 1959 to less than 5% in 2002, shown to you all in figure 21.4. That is here. And the yellow diamonds, they indicate the frequencies of melanic Biston bitularia in Michigan, and the blue squares indicate the corresponding frequencies in Pennsylvania. And then the red circles, they are those found in Great Britain, the Biston bitularia moths at Caldy Common in Great Britain. So the drop is consistent with a 15% selective disadvantage acting against moths with the dominant melanic allele. It correlates well with a significant drop in air pollution, particularly with a lowering of the levels of sulfur dioxide and suspended particles, both of which act to darken trees. Interestingly, the same reversal of melanism occurred in the United States. Of 576 peppered moths collected at a field station near Detroit from 1959 to 1961, 515 were melanic, and the frequency of 89%. The American Clean Air Act, passed in 1963, led to significant reductions in air pollution. Resampled in 2001, the frequency of melanics at the Detroit field station had also dropped to less than 5%. A similar decreasing trend was also observed in Pennsylvania, shown in the same figure as I just mentioned here and shown here. So the moth populations in Liverpool, Detroit, and Pennsylvania all part of the same natural experiment, exhibit strong evidence for natural selection. Although the evidence for natural selection in the case of the peppered moth is strong, Tutt's hypothesis that predation on color mismatched moths is the underlying mechanism of selection is currently being reevaluated. Researchers have noted that the recent selection against melanism does not appear to correlate with the changes in the abundance of light-colored tree lichens. At Caldy Common, the light form of the peppered moth began to increase in frequency long before the lichens began to reappear on trees. At the Detroit Field Station, the lichens never changed significantly as the dark moths first became dominant and then declined over a 30-year period. In fact, investigators have not been able to find peppered moths on Detroit trees at all, whether covered with lichens or not. Some evidence suggests that the moths rest on leaves in the treetops during the day, but no one is sure. Could poisoning by pollution, rather than predation by birds, be the agent of natural selection on the moths? Perhaps, but to date, only selection resulting from bird predation has been demonstrated. Researchers supporting the bird predation hypothesis point out that a bird's ability to detect moths may depend less on the presence or absence of lichens and more on other ways in which the environment has darkened by industrial evolution. Pollution tends to cover all objects in the environment with a fine layer of particulate dust, which tends to decrease how much light is reflected by the surface. In addition, pollution has a particularly severe effect on birch trees, which are light in color. Both effects would tend to make the environment appear darker, and thus would favor darker moths by protecting them from predation by birds. Despite this uncertainty over the agent of selection, the pattern is clear. Kettlewell's experiments established indisputably that selection favors dark moths in polluted habitats and light moths in pristine areas. The increase and subsequent decrease in the frequency of melanic moths correlated with the levels of pollution independently on two continents demonstrates clearly that this selection drives evolutionary change. The current reconsideration of the agent of natural selection illustrates well that the way in which scientific progress is achieved, hypotheses such that Tut's are put forth and then tested. They are rejected, new hypotheses are formulated, and the process begins anew. Humans have imposed selection upon plants and animals since the dawn of civilization. Just as in natural selection, such artificial selection operates by favoring individuals with certain phenotypic traits, allowing them to reproduce and pass their genes on to the next generation, assuming that phenotypic differences are genetically determined. This directional selection should lead to evolutionary change, and indeed it has. Artificial selection imposed in laboratory experiments, agriculture, and the domestication process has produced substantial change in almost every case in which it has been applied. This success is strong proof that selection is an effective evolutionary process. With the rise of genetics as a field of science in the 1920s and 30s, researchers began conducting experiments to test the hypothesis that selection can produce evolutionary change. A favorite subject was the laboratory fruit fly, Drosophila melanogaster. Geneticists have imposed selection on just about every conceivable aspect of the fruit fly, including body size, eye color, growth rate, lifespan, and exploratory behavior, with a consistent result. Selection for a trait leads to a strong and predictable evolutionary response. In one classic experiment, scientists selected 4 fruit flies with many bristles, stiff hair-like structures on their abdomen. At the start of the experiment, the average number of bristles was 9.5. Each generation, scientists picked out 20% of the population with the greatest number of bristles and allowed them to reproduce, thus establishing the next generation. After 86 generations of this directional selection, the average number of bristles had quadrupled to nearly 40. In another experiment, fruit flies in one population were selected for high numbers of bristles, while fruit flies in the other cage were selected for low number of bristles. Within 35 generations, the population did not overlap at all in variation, and you can see that in figure 21.25 shown to you. Similar experiments have been conducted on a wide variety of other laboratory organisms. For example, by selecting for rats that were resistant to tooth decay, in less than 20 generations, scientists were able to increase the average time for onset of decay from barely over 100 days to greater than 500 days. Familiar livestock such as cattle, pigs, and crops such as corn and strawberries are greatly different than their wild ancestors. If you look at the picture now, you can see that. Teosinte, which can be found today in a remote part of Mexico, is very similar to the ancestor of modern corn. Artificial selection has transformed it into the form we know today. And you see those two intermediates in the middle. And finally, modern day corn on the cob. So these differences have resulted from generations of human selection for desirable traits such as greater milk production and larger corn ear size. An experiment with corn demonstrates the ability of artificial selection to rapidly produce major changes in crop plants. In 1896, agricultural scientists began selecting for the oil content of corn kernels, which initially was 4.5%. Just as in the fruit fly experiments, the top 20% of all individuals were allowed to reproduce by 1986, at which time 90 generations had passed. Average oil content of the corn kernels had increased approximately 450%. Corn oil. Human-imposed selection has produced a great variety of breeds of cats, dogs, pigeons, and other domesticated animals, as you can see here. In some cases, breeds have been developed for particular purposes. Greyhound dogs, for example, resulted from selection for maximal running ability, resulting in an animal with long legs, a long tail for balance, an arched back to increase stride length, and great muscle mass. By contrast, the odd proportions of the ungainly dachshund resulted from selection for dogs that could enter narrow holes in pursuit of badgers. In other cases, varieties have been selected primarily for their appearance, such as the many colorful breeds of pigeons or cats. Domestication also has led to unintentional selection for some traits. In recent years, as part of an attempt to domesticate the silver fox, Russian scientists chose the most docile animals in each generation and allowed them to reproduce. Within 40 years, most foxes were exceptionally tame, not only allowing themselves to be petted, but also whimpering to get attention and sniffing and licking their caretakers. In many respects, they have become no different from domestic dogs. It was not only their behavior that changed, however. These foxes also began to exhibit other traits seen in some dog breeds, such as different color patterns, floppy ears, curled tails, shorter legs, and tails. Presumably, the genes responsible for docile behavior either affect these traits as well or are closely linked to the genes for these other traits. The phenomena of pleiotropy and linkage were discussed in chapters 12 and 13, respectively. And you're seeing right here what happened with the silver foxes being domesticated from the scientists. Given that we can observe the results of selection operating over a relatively short time, most scientists think that natural selection is a process responsible for evolutionary changes documented in the fossil record. Some critics of evolution accept that selection can lead to changes within a species, but contend that such changes are relatively minor in scope and not equivalent to the substantial changes documented in the fossil record. In other words, it is one thing to change the number of bristles on a fruit fly or the size of an ear of corn, and quite another to produce an entirely new species. This argument does not fully appreciate the extent of change produced by artificial selection. Consider, for example, the existing breeds of dogs, all of which have been produced in the last few thousand years after wolves were domesticated, perhaps 15,000 years ago. If the various dog breeds did not exist, and a paleontologist found fossils of animals similar to dachshunds, greyhounds, mastiffs, and chihuahuas, there is no question that they would be considered different species, and indeed the differences in size and shape exhibited by these breeds are greater than those between members of different genera in the family Canidae, such as coyotes, jackals, foxes, and wolves, which have been evolving separately for 5 to 10 million years. Consequently, the claim that artificial selection produces only minor changes is clearly incorrect. If selection, operating over a period of only a few thousand years, can produce such substantial differences, it should be powerful enough over the course of many millions of years to produce the diversity of life we see around us today. The most direct evidence that evolution has occurred is found in the fossil record. Today we have a far more complete understanding of this fossil record than was available in Darwin's time. Fossils are the preserved remains of once living organisms. They include specimens preserved in amber, Siberian permafrost, and dry caves, as well as the more common fossils preserved as rocks. Fossil rocks are created when three events occur. First, the organism must become buried in sediment, then the calcium in the bone or other hard tissue must mineralize, and finally the surrounding sediment must eventually harden to form rock. The process of fossilization occurs only rarely. Usually plant or animal remains decay or are scavenged before the process can begin. In addition, many fossils occur in rocks that are inaccessible to scientists. When they do become available, they are often destroyed by erosion or other natural processes before they can be collected. As a result, only a small fraction of the species that have ever existed, which is estimated to be about as many as over 500 million, are known from fossils. So that's not many, a small fraction. Nonetheless, the fossils that have been discovered are sufficient to provide detailed information on the course of evolution through time. By dating rocks in which fossils occur, we can get an accurate idea of how old the fossils are. In Darwin's day, rocks were dated by their position with respect to one another, called relative dating. Rocks in lower strata are generally older because young rocks form on top of the older ones. Knowing the relative positions of sedimentary rocks and the rates of erosion of different kinds of sedimentary rocks in different environments, geologists of the 19th century derived a fairly accurate idea of the relative ages of rocks. Today, geologists can determine the absolute age of rocks using isotopic dating. At the time that a rock forms, some elements exist as different isotopes. Over time, the less stable isotope is converted into the other isotope, and the ratio of the two forms changes. So, for example, potassium is one of the most common atoms in organisms. All potassium atoms have the same number of protons, but the isotopes of potassium vary in the number of neutrons they have. 40K has 19 protons and 21 neutrons and is less stable than 39K or 41K, referring to potassium, you all. 40 potassium is converted or decays over time and forms argon 40. The potassium 40 half-life is 1.25 billion years. That is, it takes 1.25 billion years for the amount of 40 potassium to decrease by 50%. The long half-life makes it useful for dating ancient fossils by determining the ratio of potassium 40 to argon 40. And you can see much of that here between the parent isotope and that daughter isotope. For events that occurred more recently, radiocarbon dating can be used. Carbon in the form of atmospheric CO2 with a mix of isotopes 14 carbon and 12 carbon is incorporated into plants via photosynthesis. The relative amount of 14 to 12 C decreases with a half-life of about 5,700 years. So other isotopes can be used for more intermediate dates. When fossils are arrayed according to their age, from oldest to youngest, they often provide evidence of the successive evolutionary change. At the largest scale, the fossil record documents the course of life through time, from the origin of prokaryotic and then eukaryotic organisms through the evolution of fishes, the rise of land-dwelling animals, and the reign of dinosaurs on the origin of humans. In addition, the fossil record shows the waxing and waning of biological diversity through time, such as the periodic mass extinctions that have reduced the number of living species. Given the low likelihood of fossil preservation and recovery, it is not surprising that there are gaps in the fossil record. Nonetheless, intermediate forms are often available to illustrate how the major transitions in life occurred. Undoubtedly, the most famous of these are the oldest known bird, Archaeopteryx, meaning ancient feather, which lived around 165 million years ago. This species is clearly intermediate between birds and dinosaurs. Its feathers are similar in many respects to those of birds today, and other features clearly reveal that it is a bird. Nonetheless, in many other respects, for example, possession of teeth, a bony tail, and other anatomical characteristics, it is indistinguishable from some carnivorous dinosaurs. Indeed, it is so similar to these dinosaurs that several specimens lacking preserved feathers were misidentified as dinosaurs and lay in the wrong natural history museum cabinet for several decades before the mistake was discovered. Archaeopteryx reveals a pattern of commonly seen intermediate fossils, rather than being intermediate in every trait. Such fossils usually exhibit some traits like their ancestors and others like their descendants. In other words, traits evolve at different rates than they're different times. So, in other words, I said traits evolve at different rates and at different times. Expecting an intermediate form to be intermediate in every trait would not be correct. The first Archaeopteryx fossil was discovered in 1859, the year Darwin published On the Origin of Species. Since then, paleontologists have continued to fill in the gaps on the fossil record. Today, the fossil record is far more complete, particularly among the vertebrates. Fossils have been found linking all the major groups. Recent years have seen spectacular discoveries, closing some of the major remaining gaps in our understanding of vertebrate evolution. For example, four-legged aquatic animals have been discovered that provide important insights concerning the evolution of whales and dolphins from land-dwelling hoof-foot ancestors. Similarly, fossil snakes with legs have shed that gradually fossil snakes with legs have shed light on the evolution of snakes, which are descended from lizards that gradually became more and more elongated with the simultaneous reduction and eventual disappearance of limbs. Tiktaalik, a species that bridged the gap between fish and the first land-living vertebrates. And then on a finer scale, evolutionary change within some types of animals is known in exceptional detail. For example, about 200 million years ago, oysters underwent a change from having small curved shells to having larger, flatter ones, with progressively flatter fossils seen in the fossil record over a period of 12 million years. A host of other examples illustrate these similar records of successive change. The demonstration of this successive change is one of the strongest lines of evolution, one of the strongest lines of evidence that evolution has occurred. And the evolution of horses is a prime example of evidence from fossils. I'll be brief here with the evolution of horses. And this shows the fossil of the Archaeopteryx, the first bird. One of the most studied cases in the fossil record concerns the evolution of horses. Modern-day members of the family Equidae includes horses, zebras, donkeys, and asses, all of which are large, long-legged, fast-running animals adapted to living on open grasslands. These species, all classified in the genus Equus, are the last living descendants of a long lineage that has produced 34 genera since its origin in the Eocene period, approximately 55 million years ago. Examination of these fossils have proved particularly well documented in the case of how evolution has proceeded through adaptation to changing environments. So the first horse, the earliest known members of the horse family species in the genus Hyracotherium, didn't look much like the modern-day horses at all. They were small, with short legs and broad feet. These species occurred in wooded habitats, where they probably browsed on leaves and herbs and escaped predators by dodging through openings in the forest vegetation. Changes in size. The first species of horses were as big as a large house cat or a medium-sized dog. By contrast, modern equids can weigh more than 500 kilograms. Examination of the fossil record reveals that horses have changed little in size for the first 30 million years. Toe reduction. Feet of modern horses have a single toe enclosed in a tough, bony hoof. By contrast, Hyracotherium had 4 toes on its front feet and 3 on its hind feet. Rather than hooves, these toes were encased in fleshy pads like those of dogs and cats. Tooth size and shape. The teeth of Hyracotherium were small and relatively simple in shape. Through time, horse teeth have increased greatly in length and have developed a complex pattern of ridges on their molars and premolars. The effect of these changes is able to produce teeth better capable of chewing tough and gritty vegetation, such as grass, which tends to wear their teeth down. As with body size, evolutionary change has not been constant through time. Rather, much of the change in the tooth shape has occurred within the past 20 million years, and changes have not been constant across all horse lineages. All these changes may be understood as adaptations to changing global climates. In particular, during the late Miocene and early Oligocene epochs, grasslands became widespread in North America, where much of the horse evolution occurred. As horses shifted from forests to grasslands, high-speed locomotion probably became more important to escape predators. By contrast, the greater flexibility provided by multiple toes and shorter limbs, which was advantageous for ducking through complex forest vegetation, was no longer beneficial. For many years, horse evolution was held up as an example of constant evolutionary change through time. Some even saw the record of horse evolution evidence for a progressive guiding force, consistently pushing evolution in a single direction toward longer limbs, fewer toes, and larger, more complex teeth. We now know that such views are misguided, and that the course of evolution changes over millions of years is rarely so simple. One reason horse evolution was originally conceived as linear through time may be that modern horse diversity is relatively limited. For this reason, it is easy to mentally picture a straight line from the Hyracotherium to modern-day Equus. But today's limited horse diversity, only one surviving genus, is unusual. In fact, at the peak of horse diversity in the Miocene epoch, 13 genera of horses could be found in North America alone. These species differed in body size and in a wide variety of other characteristics. Presumably, they lived in different habitats and exhibited different dietary preferences. Had this diversity existed to modern times, early evolutionary biologists would likely have had a different outlook on horse evolution. Much of the power of the theory of evolution is its ability to provide a sensible framework for understanding the diversity of life. Many observations from throughout biology simply cannot be understood in a meaningful way except as a result of evolution. As vertebrates have evolved, the same bones have sometimes been put to different uses, yet the bones are still recognizable. their presence betraying their evolutionary past. For example, the forelimbs of vertebrates are all homologous structures with different appearances and functions that all derive from the same body part and a common ancestor. If you look at figure 21.13, you can see how the bones in the forelimb have been modified in different ways for different animals. Why should these very different structures be composed of the same bones? A single upper forearm bone, a pair of lower forearm bones, and several small carpals and one or more digits. If evolution had not occurred, this would indeed be a riddle. But when we consider all of these animals are descended from a common ancestor, it is easy to understand that natural selection has modified the same initial starting blocks to serve very different purposes. Some of the strongest anatomical evidence supporting evolution comes from comparisons of how organisms develop. Embryos of different types of vertebrates, for example, often are similar early on, but become more different as they develop. Early in their development, vertebrate embryos possess pharyngeal pouches or pharyngeal gill slits, which develop into different structures. In humans, for example, they become various glands and ducts. In fish, they turn into gills. At a later stage, all primate embryos have a long tail. But whereas monkeys and most primates keep the tail, all we and our ape relatives retain is the coccyx at the end of our spine. Human fetuses even possess a fine fur called lanugo during the 5th month of development. Similarly, although frogs go through a tadpole stage, some species develop directly and hatch out as little fully formed frogs. However, the embryos of these species still exhibit tadpole features, such as the presence of a tail, which disappear before the froglet hatches. Because natural selection can work only on the variation present in a population, it should not be surprising that some organisms do not appear perfectly adapted to their environments. For example, most animals with long necks have many neck vertebrae for enhancing flexibility. Geese have up to 25, and plesiosaurs, the long-necked reptiles that patrolled the seas during the age of dinosaurs, had as many as 76. By contrast, giraffes have only 7 very long neck vertebrae. Why haven't they evolved more, like other long-necked animals? It turns out that almost all mammals have only 7 neck vertebrae. Because mammal species have no variation in vertebra number among individuals in a population, natural selection has nothing to work with, and thus there is no way to lead to an increased number of vertebrae. In the absence of variation in vertebra number, selection led to an evolutionary increase in vertebra size to produce the long-necked giraffe. An excellent example of imperfect design is the eye of vertebrate animals, in which photoreceptors face backward toward the eye wall or the wall of the eye, shown to you all in figure 2115a. And as a result, the nerve fibers extend not backward toward the brain, but forward into the eye chamber where they slightly obstruct light. Moreover, these fibers bundle together to form the optic nerve, which exits through a hole at the back of the eye, creating a blind spot. By contrast, the eyes of mollusks, such as squid and octopuses, are better arranged. The photoreceptors face forward, and the nerve fibers exist at the back, neither obstructing light nor creating a blind spot. Such examples illustrate that natural selection is like a tinkerer, working with whatever material is available to craft a workable solution, rather than like an engineer who can design and build the best possible structure for a given task. Workable but imperfect structures, such as a vertebrate eye, are an expected outcome of evolution. Many organisms possess vestigial structures. Vestigial structures that have no apparent function, but resemble structures that their ancestors possessed. Humans, for example, possess a complete set of muscles for wiggling their ears, just as many other animals do. Although these muscles allow other animals to move their ears to pinpoint sound, such as the movements or growl of a predator, they have little purpose in humans other than amusement. As other examples, boa constrictors have hip bones and rudimentary hind legs. Manatees, a type of aquatic animal often referred to as sea cows, have fingernails on their flippers, which evolved from legs. Some blind cavefish, which never see the light of day, have small, non-functional eyes. The human vermiform appendix has long thought to be vestigial. It represents the degenerate terminal part of the cecum, the blind pouch or sac in which the large intestine begins. In other animals, such as mice, the cecum is the largest part of the large intestine and functions in storage, usually bulk cellulose in herbivores. Although greatly diminished in size, some recent evidence suggests that the human appendix may not be vestigial because it may harbor beneficial intestinal bacteria. Vestigial or not, the human appendix can be a dangerous organ. Appendicitis, which results from an infection of the appendix, can be fatal. Vestigial traits can also be seen in genomes of many organisms. For example, the ice fish is a bizarre-looking, nearly see-through fish that lives in the frigid waters of the Antarctic. The ice fish's transparency results not only from a lack of pigment in its body structures, but also from the near invisibility of its blood. Our blood is red due to the presence of red blood cells, which contain hemoglobin, the molecule that transports oxygen from the lungs to the tissues. However, oxygen concentration in water increases as the temperature decreases. The waters of the Antarctic, which are about 0 degrees Celsius, contain so much oxygen that the fish do not need special molecules to carry oxygen. The result is that the fish do not have hemoglobin, and consequently, their blood is colorless. Nonetheless, when the DNA of the ice fish was examined, scientists discovered that they have the same gene that produces hemoglobin as that found in other vertebrates. However, the ice fish hemoglobin gene has a variety of mutations that render it non-functional, and thus the ice fish does not produce hemoglobin. The presence of this inoperable version of the hemoglobin gene in the ice fish means that its ancestors had hemoglobin. However, once this fish's progenitors occupied the cold waters of the Antarctic and lost the need for hemoglobin, mutations that would prevent the production of hemoglobin and thus would be filtered out by natural selection in other species were able to persist in the population. Just by chance, some of these mutations increased in frequency in the population through time, eventually becoming established in all individuals and knocking out the fish's ability to produce hemoglobin. Pseudogenes, sometimes called fossil genes, because they are traces of previously functioning genes, such as the hemoglobin in the ice fish, are actually quite common in the genomes of most organisms. When a trait disappears, the gene does not just vanish from the genome. Rather, some mutation renders it inactive, and once that occurs, other mutations can accumulate. It is difficult to understand vestigial structures such as these as anything other than evolutionary relics, holdovers from the past. However, the existence of vestigial structures argues strongly for the common ancestry of the members of the groups that share them, regardless of how different those groups have subsequently become. All of these anatomical lines of evidence, homology, development, and imperfect and vestigial structures are readily understandable as a result of descent with modification. That is evolution. Fingernails of the West Indian manatee and then the pseudogenes, which I just mentioned there. And this shows the ice fish. Biogeography is the study of the geographic distribution of species. It reveals that different geographic areas sometimes exhibit groups of plants and animals of strikingly similar appearance, even though the organisms may only be distantly related. It is difficult to explain so many similarities as a result of coincidence. Instead, natural selection appears to have favored parallel evolutionary adaptations in similar environments, because selection in these instances has tended to favor changes that made the two groups more alike than their phenotypes have converged. This form of evolutionary change is referred to as convergent evolution. The best known case of convergent evolution, in the best known case of convergent evolution, two major groups of mammals, marsupials and placentals, have evolved in very similar ways in different parts of the world. Marsupials are a group in which the young are born in a very immature condition and held in a pouch until they are ready to emerge into the outside world. In placentals, by contrast, offspring are not born until they can safely survive in the external environment with varying degrees of parental care. Australia separated from the other continents more than 70 million years ago. At that time, both marsupial and placental mammals had evolved, but in different places. In particular, only marsupials occurred in Australia. As a result of this separation, the only placental mammals in Australia today are bats and a few colonizing rodents, which arrived relatively recently, and the continent is dominated by marsupials. What are the Australian marsupials like? To an astonishing degree, they resemble the placental mammals living today on the other continents. Please see Figure 21.18, shown for you here now. The similarity between some individual members of these two sets of mammals argues strongly that they are the result of convergent evolution, similar forms having evolved in different isolated areas because of similar selective pressure in similar environments. When species interact with the environment in similar ways, they are often exposed to similar selective pressures, and they therefore frequently develop the same evolutionary adaptations. Consider, for example, fast-moving marine predators, such as those we see here. This is Figure 21.19, or 21.18, excuse me. The hydrodynamics of moving through the water require a streamlined body shape to minimize friction. It is no coincidence that dolphins, sharks, and tuna are among the fastest of marine species, have all evolved to have the same basic shape. We can infer, as well as the ichthyosaurs, marine reptiles that lived during the age of dinosaurs, exhibited a similar lifestyle. Island trees exhibit a similar phenomenon. Most islands are covered by trees, or were until the arrival of humans. Careful inspection of these trees, however, reveals that they are not closely related to the trees with which we are familiar. Although they have all the characteristics of trees, such as being tall and having a tough outer covering, in many cases, island trees are members of plant families that elsewhere exist only as flowers, shrubs, or other small bushes. For example, on many islands, the native trees are members of the sunflower family, Asteraceae. Why do these plants evolve into trees on islands? Probably because the seeds from the trees rarely make it to isolated islands. As a result, those species that do manage to colonize distant islands face an empty ecological landscape upon arrival. In the absence of other tree-like plants, natural selection often favors individual plants that can capture the most sunlight for photosynthesis, and the result is the evolution of tree-like forms on islands throughout the world. Convergent evolution is even seen in humans. People in most populations stop producing lactase, the enzyme that digests milk, sometime in childhood. However, individuals in African and European populations that raise cattle produce lactase throughout their lives. DNA analysis indicates that the retention of lactase production into adulthood is a result of different mutations in Africa and Europe, which indicates that the populations have independently, convergently acquired this adaptation. And to end things in the chapter, Darwin made several important observations during his voyage around the world. He noted that islands are often missing plants and animals common on continents, such as frogs and land mammals. Accidental human introductions have proved that these species can survive if they are released on islands, so lack of suitable habitat is not the case. In addition, those species that are present on islands often have diverged from their continental relatives, and sometimes, as Darwin's finches and the island trees just discussed, occupy ecological niches used by other species on continents. Lastly, island species usually are more closely related to species on nearby continents, even though the environment on the continents and nearby islands often is not very similar. Darwin deduced the explanation for these phenomena. Many islands have never been connected to continental areas. Many species that occurred there arrived there by dispersing across water. Some species, those that can fly, float, or swim, are more likely to get to the island than others. Some, like frogs, are particularly vulnerable to dehydration in salt water and have almost no chance of island colonization. And lastly, the absence of some types of plants and animals provides opportunity to those that do arrive. As a result, colonizers, which usually come from nearby areas, often evolved into many species exhibiting great ecological and morphological diversity. This term, adaptive radiation, will be discussed in Chapter 22, our next chapter. So, given this, Darwin also had critics. In the century and a half since he proposed it, Darwin's theory of evolution by natural selection has become nearly universally accepted by biologists, but has been a source of controversy among some members of the general public. Though I will not discuss those here, it is found in the last section of Chapter 21, and you can see those things noted here. This has been your instructor, Scholar Huff. Thank you all.